Today's question is, a transmembrane receptor protein X and a transmembrane protein bound to the actin cytoskeleton Y with fluorescent tags were expressed in a cell. A fluorescence recovery after photobleaching or FRAP experiment was performed on these proteins. Which one of the following options represents the most likely outcome of this experiment? Now, we have been given 4 options here. First, let's understand what FRAP is. Fluorescence recovery after photobleaching or FRAP is a microscopy-based technique used to study the mobility and diffusion of proteins and other molecules within cells. It involves bleaching a specific area of a fluorescently labeled sample, for example, GFP labeled proteins, with a high-intensity laser, and then monitoring the recovery of fluorescence in that area over time using a lower intensity laser. The rate and extent of fluorescence recovery provide information about the mobility and dynamics of the molecules being studied. Overall, FRAP provides valuable insights into the dynamic nature of cells and their components, helping researchers understand how proteins move, interact, and perform their functions within a living cell. Now here, the option 1 is fluorescence of both proteins will be recovered at the same rate. The rate of fluorescence recovery in FRAP is directly related to the mobility of the fluorescent molecule. More mobile molecules can diffuse back into the bleached area faster, leading to a quicker fluorescence recovery. This shows the positions of the transmembrane receptor protein X and the transmembrane protein Y bound to the actin cytoskeleton before photobleaching. Although the fluorescence of proteins X and Y can be recovered, these proteins will have different mobilities due to differences in their interactions with the cellular environment. Therefore, this option is incorrect. Now, the second option is fluorescence of X will be recovered later than Y. X is a freely diffusing transmembrane protein not bound to the cytoskeleton. Thus, it will have a high degree of mobility, resulting in a fast fluorescence recovery. Overall, since X is more mobile, it will recover faster than Y. Therefore, this option is incorrect. Next, the fourth option is, being membrane proteins, no recovery of fluorescence is expected. Although membrane proteins are generally confined to the cell membrane, they can still exhibit lateral diffusion within the membrane, allowing for fluorescence recovery. Therefore, this option is incorrect. The third option is fluorescence of Y will be recovered later than X. If protein Y is bound to the actin cytoskeleton, its mobility will be significantly restricted, as the cytoskeleton acts as a structural framework that limits its movement. The slow movement will lead to a slower fluorescence recovery rate of protein Y compared to that of the protein X. Therefore, the most likely outcome is the third option. Fluorescence of Y will be recovered later than X. Do visit my YouTube channel Simply Biology with Manisha for more such videos. Thank you.